Today, let's see the diseases of crucifers. Club root or finger and toe disease. Causal organism. Today, let's see the diseases of crucifers. Today, let's see the diseases of crucifer. Club root or finger or toe disease. Causal organism. Plasmodiaphora brassica. Fairly severe in hilly region on many plants. Once infected, fields become unfit for cultivation for several years. Symptoms: infected plants have pale green to yellowish leaves initially; later, infected plants show wilting in the middle of hot, sunny days, recovering during the night. Stunting of above-ground parts and reduction in size of heads. Characteristic symptoms become apparent in advanced stages of root infection as spindle-like, spherical, knobby, or club-shaped swelling. The swellings may be few, or they coalesce and cover the entire root system. Survival and spread. Primary: soil-borne resisting spores which survive for longer periods in soil, up to 10 years. Secondary: autonomous zoophores, also carried through irrigation water or by root contact. Favorable condition: it occurs at temperature ranging between 12 degrees Celsius to 27 degrees Celsius. High soil moisture, neutral to acidic soil, having the pH in the range of 5 to 7. Management: use of seedling from disease-free fields. Plant cabbage and other susceptible crops in well-drained field that have a pH slightly above neutral, usually about pH 7.2. Add hydrated lime to soil to increase pH to 7.2. This is to be done 6 weeks before planting at the rate of 2.5 ton per hectare. Avoid excess irrigation. Treat the soil of seedbed areas with chlorobecrin, methyl bromide, or Ripam 2 weeks before sowing. Downy mildew. Causal organism: Peronospora parasiticum. Symptoms: small purplish brown spots on undersurface of leaves. Pale, small, yellow, angular spots on upper surface of leaves. There is also downy growth on the undersurface. The spots coalesces and the leaves shrivels and dries up prematurely. In some, these spots expose the heads of soft rod. Birds look brownish at the top. Stem show dark brown and depressed lesions or streaks which later develop downy growth of fungus. Survival and spread. Primary: use spores in infected plant debris or in soil. Secondary: wind-borne and rain-splashed sporangium. Favorable conditions: it occurs at a moderate temperature range of 12 degrees to 27 degrees Celsius. High soil moisture, neutral to acidic soils having the pH range of 5 to 7. Management: Destruction of infected plant debris, avoidance of thick sowing and excessive moist conditions. Spray metalaxyl plus mancozeb redomin at the rate of 0.2% or copper oxychloride 0.3% at 10 days interval. Powdery mildew, causal organism, Erysipelas polygonia. Symptoms: white powdery spots on the upper surface of leaves, stems, flower parts, etc. Finally, the mildew may cover the entire surface. Infected plants may show rotting and do not have normal growth. Survival and spread. Primary: dormant mycelium in infected plant debris. Secondary: wind-borne conidia. Favorable conditions. It occurs at a moderate temperature range of 12 to 27 degrees Celsius. Dry conditions with morning moderate relative humidity of 80%. Management: destruction of infected plant debris, spraying of Dinocap at 0.2%. Alternaria leaf spot, causal organism, Alternaria brassicola, Alternaria brassicea, Alternaria refani. Symptoms: symptoms usually are on older leaves. Spots are small, dark colored. They enlarge and soon become circular. Under humid conditions, group of conidiophores will be formed in the spot. Spots develop concentric rings. अरे भैया रिकॉर्डिंग चल रही है, उसको कह रहा हूँ मैं. सर्वाइवल एंड स्प्रेड. प्राइमरी: अब वापस कर लो उससे और मैं आपकी एडिटिंग करवा दूंगा. मेरे भी तो हो गई है 18 मिनट की. मैं एडिट कर दूंगा. लिख लो पेज पे कौन सा नंबर कितने मिनट पे. सर्वाइवल एंड स्प्रेड. Primary: dormant mycelium in infected plant debris. Secondary: wind-borne conidia. Favorable conditions: it occurs at a moderate temperature range of 12 to 27 degrees Celsius. Dry conditions with morning relative humidity of 80%. Management: destruction of infected plant debris and spraying of Dinocap at the rate 0.2%. Alternaria leaf spot, causal organism, Alternaria brassicola, Alternaria brassicae, Alternaria refani. Symptoms usually are on older leaves. Spots are small, dark colored. They enlarge, soon become circular. Under humid conditions, group of conidiophores will be formed in the spot. Spots develop concentric rings. Finally, the spots coalesces, leading to lightening of leaves. Short poles may be formed due to falling off of infected spots tissue. The fungus is seed-borne and cause shriveling of seeds and poor germination. Linear spots also appear on petioles, stems, and pods. Survival and spread. Primary: dormant mycelium or conidia on seed or in infected plant debris. Wind-borne gonorrhea is second. Favorable conditions includes relative humidity of more than 80%, less vigorous plants, and those which are grown late in the season. Management: hot water treatment at 50 degrees Celsius for 30 minutes. Seed treatment with thiram or mancozeb. Foliar spray with mancozeb 0.25% or copper oxychloride at the rate of 0.2% twice. White rust, Albugo candida. Symptoms: white, shiny raised blisters, pustules on the lower surface of leaves, stems, and flowers. Pustules coalesces to form irregular patches. The epidermis ruptures, exposing white spore masses, which give the pustule a powdery appearance. Distortion of the floral parts, including petals, pistil, and anthers, due to hypertrophy and hyperplasia. Plants malform beyond recognition. Survival and spread. Primary. Two spores in soil and sporangia form perennial weed host in the vicinity. Secondary. Wind-borne and rain-splashed conidia, sporangia, or autonomous zoospores. Favorable conditions includes high relative humidity of more than 90%, high soil moisture, and frequent rains. Management: crop rotation, destruction of weeds, and spraying of 0.8% of podium mixture or copper oxychloride at the rate of 0.3%. Flatland, Otriodipodia theobromine. It was first reported from Maharashtra and Gujarat. It is now prevalent in all growing areas of the country. The disease is a problem in orchids above 10 years of age. Symptoms: branches become flat and twisted at the terminus, leaves become thin, small, and yellow, and are produced in clusters on the affected branches. Very few flowers are produced on affected branches which will remain infertile. Fruit set is rare, and when set fruits are undersized, hard, and fail to ripen. Foliage and fruits fall prematurely. Survival and spread. Primary: Conidia on infected plants. Secondary: wind-borne conidia. Management: pruning of affected branches followed by copper oxychloride or captan or cyanide 0.3% spray. Leaf spot Pheophyllosophora indica, Staphylococcus versicolor. Disease symptoms Pheophyllosophora indica. Earlier circular spots with pinkish then gradually to brownish in color, and the center of the spot sometimes whitish gray color, and number of spots are more on leaves. Staphylococcus versicolor spots are circular and brownish and bigger. Later stages can see the black dots on center of the spots. These black dots are the asexual fruiting body of the fungus Acervillus. In advanced stages, this leads to defoliation. Numerous small, circular, pinkish to reddish brown, conspicuous spots with whitish center on mature leaves. Spots coalesce and leaves drop prematurely. Survival and spread. The pathogen is seed-borne fungus, and inoculum present in the seeds are the source of primary infection. Fungus also survives on clones, fruits, and plant debris. Favorable condition includes rainy seasons favor the development of disease. Management: carbendazim 0.1% and companion, the combo product, includes carbendazim and mancozeb 12% and 72% to avoid resistant development in pathogen. Sooty mold Capmodium versicolor. Disease symptoms: it is a fungal disease developed on honeydew-like excretion secreted by aphid and scale insects. Disease severity increases in increased population of leafhoppers, aphids, and other insects. Black superficial growth on entire surface of leaves, fruits, and twigs. Fungus is not a parasite. It grows on the excreta and honey secretions of insects. The fungus slowly covers the entire leaf area, severely affecting the process of photosynthesis. Under dry spells, such affected leaves curl and shrivel. During flowering time, the appearance of the disease results in reduced fruit scent. The sooty mass, is a superficial growth on the fungus, and it multiplies on insect secretion. This results in reduced translocation of food to the fruit, which reduce which leads to reduction in their size. The impact of this disease is on the host photosynthetic activity, and therefore the yield decreases. Survival and spread. The severity of infection depends on the honeydew secretions by the scale insects that provides the necessary medium for the fungal growth. This transmission occurs by airborne ascospores. Favorable conditions, high humidity and moist situation favors the development of disease. Management. Spray of wettable sulfur 0.2% along with insecticide dimethoate 1.5 grams per liter. Spray of 1% starch solution makes flakes of the fungus and due to small wind falls from the plant. प्रोब्स मंजू मैम प्लीज राहुल को फोन कर दीजिए papaya ring spot virus is not spread by either insects and it does not survive in soil or dead plant material उसका अभी फोन आया था the virus can also be spread by the movement of infected papaya plants and cucurbit seedling. Once infected, plants cannot be cured by spraying with pesticides or removing plant parts showing symptoms. Management. Non-chemical control efforts to control ring spots in have included rousing, breeding, or tolerance to PRVP cultural practices. इसकी माँ की आँख मैं नहीं कर रहा हूँ मजाक बना रखा है Foot rot, stem rot, Pythium ephreni dermatum. Symptoms: water-soaked patches develop on the stem at the ground level. Patches enlarge and diseased tissue turn brown and rot. Because of rotting, the basal portion of stem is girdled, affecting the upward flow of water and nutrients and weakening the plant. Internal tissue of bark appear dry and give honeycomb appearance. Infection spreads to roots and causes decay. Terminal leaves turn yellow, wither and droop. Fruits shrivel and drop off. Entire plant topples over and die. Pathogen. Mycelium is septate, brown, and much branched. The sclerotia are black, spherical to irregular shape and produced in abundance. Survival and spread. Primary. Oospores and saprophytic mycelium in soil and infected seedlings transplanted in the main field. Secondary. Zoospore through irrigation water. Favorable conditions appears from June to August. Younger seedlings are more susceptible than older ones. Severity increases with intensity of rainfall. High soil moisture. Management. Seedlings should be raised in well-drained nursery area or in portrays in modern nurseries in protected polyhouses. Uproot the diseased seedlings and burn. Drench the base of stem with COC at the rate of 0.25% or metalaxyl plus mancozeb at the rate of 0.1% or rhodium mixture at the rate of 1%. Mosaic virus economic importance. In India, first reported from Mumbai and Pune and occurs more in central India. 90 to 100% damage occurs in severe cases. Virus is a virus with long flexus rod-shaped particles having single-stranded RNA as genome. Symptoms: mottling and puckering of leaves, especially the young ones. Water-soaked chlorotic spots on tender stem and petioles. The lamina is reduced and malformed and are often modified into tendrils-like structures giving a shoestring appearance. Decline and marked reduction in growth of diseased plants. Older leaves fall down and a small tuft of younger leaves is left at the top in upright position. Fruits develop innumerable circular water-soaked spots with concentric rings. Fruit size is severely reduced with deformed shape. Pathogen. The virus particles are rod-shaped and thermal inactivation point of the virus lies between 54 degrees Celsius and 60 degrees Celsius. Survival and spread. Primary. Virus particles on affected plant. Secondary. Aphids, namely Myzus persicae and other species, Apis malvae, Apis gossypi, Apis medicagenes, Rhopalosiphum mides, Microcyphum solanifoli. Other hosts include Cucumis sativus, Cucurbita pepo, Cucurbita maxima, Citrullus vulgaris, Nufa acutangula, Echinacea, Caesarea, safflower, and some ornamental plants. Management: disease-free seedlings raised in modern insect-free net house or polyhouses. Housing and destruction of diseased plants. Raising barrier crops like sorghum in the boundaries of the field. Vector controls with systemic insecticides like dimethoate or methyl as dimeton. Perica cauliflora is resistant. Growing transgenic protein mediated varieties like Sunup and Rin. Leaf curl. Tobacco leaf curl virus. or Nicotina virus 10. Virus is a Gemini virus with geminate spherical particles having a single-stranded DNA as gene. Symptoms: severe curling, wrinkling, and distortion of leaves, accompanied by vein tearing and reduction of leaf lamina. Leaf margins are rolled downwards and inwards in the form of inverted cup. Curled leaves have thickened veins. Leaves become leathery, brittle, and petioles are twisted. Diseased plant fail to flower or bear any fruits. In advanced stage, defoliation takes place and growth is arrested. Survival and spread. Primary. Virus particles on affected plants. Secondary. Whitefly, Nemesia typica. Virus also infect tobacco, tomato, sunhemp, chili, petunia, zinnia, datura, stramonium, hollyhock, kale, gooseberry, etc. Management: disease-free seedlings, rousing and destruction of diseased plants and other host species, erecting yellow sticky traps for whitefly trapping, vector control with monocototropes at 1.6 ml per liter or dimethoate or metacystox at 2 ml per liter. Papaya ring spot virus. The earliest symptoms on papaya are a yellowing and vein clearing of young leaves. These are followed by very conspicuous yellow mottling. Biology: these viruses typically have long flexus rod-shaped particles about 800 to 900 nanometer long and are transmitted by numerous species of aphids in a non-persistent manner. Ring spot virus is grouped into two types, PRVP and PRVW. PRVW causes major damage and was previously referred as watermelon mosaic virus also. Spread: the virus is spread from plant to plant by any species of aphids in non-persistent manner. Many species of aphids are capable of transmitting the virus, and it takes only a few seconds of feeding time for an aphid to acquire the virus into its mouthpart. It is then able to spread the virus to other plants during brief feeding. Septoria leaf spot. Causal organism: Septoria glycopersicum. Symptoms: less vigorous plants are usually affected. Small, round to irregular spots with a gray center and dark margins on leaves. Spots usually start on lower leaves and gradually advance upwards. Sporulae and leaves are blighted. Complete defoliation of affected leaves. Stems and flower are sometimes attacked. Fruits are rarely attacked. Survival and spread. Primary: mycelium or conidia in epidia in infected plant debris or also weeds. Secondary: conidia through rain, splash, or wind, and also by slimy conidia sticking onto hands and clothing of pickers. Favorable conditions: poor vigor of plant due to nutrient inadequacy or in late season, high humidity or persistent dews at 25 degrees Celsius, moist weather with intermittent showers. Management: crop rotation with non-hosts, destroy infected crop debris, ST with mancozeb or Zineb at 2 gram per kg seed, spray Zineb at the rate 0.2% or mancozeb at the rate 0.2% at 10-day interval. Line PI 422397 is resistant. Amping off. Causal organism: Pythium apnea dermatom. Symptoms: amping off occurs in two stages, that is, the pre-emergence and the post-emergence phase. In the pre-emergence the phase, the seedlings are killed just before they reach soil surface. The young radicals and the pubules are killed, and there is complete rotting of the seedlings. The post-emergence phase, characterized by the infection of the young juvenile tissue of the collar at the ground level. The infected tissues become soft and water-soaked. The seedling topples over or collapses. Mode of spread and survival. All the causal organisms are soil inhabitants and they build up in soil with the available host. Generally, these pathogens have wide host range. Management: used raised seed bed, provide light but frequent irrigation for better drainage. Drench with copper oxychloride 0.2% or water mixture 1%. Seed treatment with fungal culture Trichoderma viride 4 gram per kg of seed or Thiram 0.3 grams per kg of seeds is the only preventive measure to control the pre-emergence damping off. Spraying 0.2% metalaxyl when there is cloudy weather. Early blight. Causal organism: Alternaria solani. This is a common disease of occurring on the foliage at any stage of the growth. The fungus attacks the foliage, causing characteristic leaf spots and blight. Early blight is first observed on the plants as small black lesions, mostly on the older foliage. Spots enlarge, and by the time they are 1/4 inch in diameter or larger, concentric rings in the bull's-eye pattern can be seen in the center of the diseased area. Tissue surrounding the spots may turn yellow. If high temperature and humidity occur at this time, much of the foliage is killed. Lesions on the stems are similar to those on leaves, sometimes girdling the plant if they occur near the soil line. The fungus also infests the fruit, generally through the calyx or stem attachment. Lesions attain considerable size, usually involving nearly the entire fruit. Concentric rings are also present on the fruit. Survival and spread. Primary: the fungus spends The winter in infected plant debris in or on the soil where it can survive at least one and perhaps several years. It can also be seed-borne. Secondary. The spores are transported by water, wind, insects, other animals including man and machinery. Favorable conditions includes warm, rainy and wet weather. Management, removal and destruction of crop debris. Practicing crop rotation helps to minimize the disease incidence. Spraying the crop with mancozeb 0.2% for effective disease control. Leaf curls. Causal organism, leaf curl virus (LCV). Symptoms: leaf curl disease is characterized by severe stunting of the plant with downward rolling and crinkling of the leaves. The newly emerging leaves exhibit slight yellow coloration and later they also show curling symptoms. Older leaves becomes leathery and brittle. The nodes and internodes are significantly reduced in size. The infected plants look pale and produce more lateral branches, giving a bushy appearance. Mode of spread and survival is neither seed nor subtransmissible, but seed from fresh fruit having infection may have the virus on the seed pod. The virus is transmitted by whiteflies, Yersinia typici, and grafting. Even a single virulliferous insect is able to transmit the virus. Favorable conditions. Whitefly is the vector for transmitting of leaf curl virus. Management. Keep yellow sticky traps at the rate 12 per hectare to monitor the whitefly. Raise barrier crops, cereals around the field. Removal of weed host. Protected nursery in net house or greenhouse. Spray imidacloprid 0.05% or dimethoate 0.05% at the rate of 15, 25, 45 days after transplanting to control the vector. Mosaic, Mosaic virus. Symptoms. The disease is characterized by light and dark green mottling on the leaves, often accompanied by wilting of young leaves in sunny days when plants first become infected. The leaflets of affected leaves are usually distorted, puckered, and smaller than normal. The affected plant appears stunted, pale green, and spindly. Survival and spread. The virus is spread by contact with clothes, hands of working labor, touching of infected plant with healthy ones, plant debris, and plant roots and implements. Seed from management. Seeds from disease-free, healthy plants should be selected for sowing. Soaking of the seeds in a solution of praseodymium phosphate 90 grams per liter of water a day before sowing. Seeds should be thoroughly rinsed and dried in shade. In the nursery, all the infected plants should be removed carefully and destroyed. Crop rotation with crops other than tobacco, potato, chili, capsicum, brinjal, should be undertaken.